Hello, and welcome back to the chapter on enzymes. So in the previous chapter or in the previous video, we have talked about some examples of enzymes, the definition of an enzyme, and how enzymes are named. So let's continue and talk about the characteristics of enzymes. Okay, there are two learning outcomes. Firstly, explain the mode of action of enzymes in terms of an active site, enzyme substrate complex, lowering of activation of energy, and enzyme specificity using the lock and key hypothesis. Investigate and explain the effects of temperature and pH on the rate of enzyme catalyzed reactions. Okay, so we have watched a little video about this detergent, right? And this particular enzyme-based biological detergent, honestly, is not very common. I also personally use chemical-based detergent because biological-based detergents actually need a certain temperature to work well. Okay, and that's not always possible sometimes. So enzymes are very highly specific in their action. For example, amylase only acts on starch and not on proteins or fats. Similarly, proteases act only on proteins and lipase only on fats, right? Also known as lipids. This enzyme specificity means that each chemical reaction inside a cell is catalyzed by a unique enzyme. Now inside our body, there are actually hundreds of different enzymes. Each of them have a certain purpose and they only do that one thing. And that is what I mean by highly specific. A amylase cannot act on other things. A lipase only acts on fat and nothing else. Okay, why is that the case? The reason why there's this thing called enzyme specificity is because of the theory or the model of the lock and key hypothesis. So we know that enzymes are made from proteins and they have a three-dimensional shape. So this lock and key hypothesis explains how the shape of an enzyme affects the way it functions. Okay, you may recall that in chemistry, you often talk about this thing called reactants and products, right? So there's also a similar concept in this, and we'll call the reactants we can call it as a substrate also, where only in examples of enzymes, then we call it as substrates. Okay, so the substances on which an enzyme will act on are called substrates. For example, protein is the substrate on which a protease acts. According to the lock and key hypothesis, enzyme reactions depend on the presence of active sites. Active sites are grooves or pockets on the surface of an enzyme molecule into which the substrate molecule with the matching shape can fit, just like a lock and key. The enzyme is the lock and the substrate is the key. Alright, so let's look at this picture over here. This is a picture of a pepsin enzyme. In the pepsin enzyme, there's a specific area of it that is called the active site. And this active site has a specific shape. Right? In fact, it's a specific three-dimensional shape because if you rotate it around, you'll see that there's a certain 3D shape of it. So what happens is that when the substrate happens to fit into this active site, it will then form what I call as an enzyme substrate complex. Okay, and only that specific substrate, which in this case is the protein, right? The protein is able to fit into this area. Nothing else can fit. And the starch cannot fit, the lipids cannot fit because they do not have the same shape. Okay, so we don't, you all have a lock, right? Those traditional locks, those older version of locks, right, where they has to put a key inside, not the nowadays a modern lock with press button one, right? And you know that each lock only can be opened by a specific key. So it's the same. The only issue with this hypothesis, right, that some would complain about this, is that in this case, it is the key that actually changes and not the lock. The lock is an enzyme, right? So in a traditional lock, you put the key in, you turn, then the lock will pop open, right? But in this case, it's not the way, right? What's happening is that the key is the thing that actually changes into your products at the end. But the reason why they use this lock and key hypothesis is because of the specificity, right? Only one key to one lock. Okay. All right. So when the substrate binds to the active site of the enzyme, an enzyme substrate complex is formed. This is a temporary molecule that forms when substrates bind to the enzyme. Reactions take place at the active site to convert the substrate molecule into product molecules. The product molecules separate from the enzyme, and the enzyme molecule remains unchanged and is free to combine again with more substrate molecules. Okay, so this also tells us that the enzyme is unchanged. It does not it's not used up in the reaction, so you can continue to do more reactions with it. Okay, there's this little equation here. Enzyme plus substrate forms enzyme substrate complex, and then it breaks down the substrate into the products. So we got the enzyme and the products. Now this is a very important page over here, so please put a big star beside it. You will be required to memorize more or less everything in this page. I have a certain formula where I will teach you of how to remember this, but for now let's just read it first. Okay, this is how the lock and key hypothesis works, or how to use the lock and key hypothesis to explain any enzyme reaction. Okay, so an enzyme has a specific three-dimensional shape. It has a depression called the active site. The substance on which the enzyme acts is called the substrate. Okay, so as we can see here, this is a 2D version, but you keep it bear in mind that it's actually 3D. So this is the substrate, right, and this is the enzyme. Only the substrate with a 3D shape complementary to the active site can fit into the enzyme. So the word complementary means that is the same shape. Okay, then this results in the formation of an enzyme substrate complex. Okay, I will start to say ESC when I teach you or in the lesson, but for exams you cannot write ESC. You have to write the whole thing called enzyme substrate complex, but saying it out is a bit of a mouthful, so I may start to say ESC once in a while. Okay, just be aware of that. Alright, so the enzyme is just like a lock and the substrate is the key. The substrate fits into the enzyme just like a key fits into a lock. This is called the lock and key hypothesis. Okay, so while the substrate is attached to the active site, a chemical reaction occurs. The substrate is converted to the products. Okay, so actually what happens is that in the active site, there are certain molecules or certain certain parts of it that will cause a chemical reaction, and then it would cleave the substrate into its products. Okay, I can make it simple to say it breaks down the substrate into its products. So yeah, there's a chemical reaction that occurs. Okay, then later the products leave the active site and the enzyme remains unchanged. Okay, some teachers will say the enzyme remains chemically unchanged also. It can catalyze another reaction. So this is the final product. Okay, so they also teach you the word complementary is like matching. Okay, just want to talk a bit more about this lock and key hypothesis, because this is actually one of the most important parts for this chapter. So I will have a certain method where I use to recall it. I call it the L and K, SAC, ESC, SPU method. Okay, so it's L and K, sac, escape, spool. Okay, it's a bit stupid sounding, but that's how I remember. So I always start by talking about the lock and key. Okay, so what I mean is that let's say I have any enzyme or any substrate. I must be specific with them. I can't just say this enzyme or that enzyme. I'm going to say the name of the enzyme. Okay, so let's say for example, I'm going to talk about amylase in this case. Okay, so I'll start by saying L and K. L and K stands for lock and key. So I will say the amylase is the lock and the starch is the key. Okay, the amylase of course is the enzyme, the starch is the substrate. Then I'm going to talk about this thing called SAC. S stands for specific three-dimensional shape. Okay, so then I'll say the enzyme has a specific 3-dimensional shape. A stands for active site. I'll say the enzyme has a active site. S stands for specific. Okay, and I said that a few times, right? SAC. Oh, sorry. The C stands for complementary. The C stands for complementary. So I'll say the substrate, which is starch, is complementary to the active site of the amylase. Okay, so then I'll continue on to say ESC. ESC stands for enzyme substrate complex. So I'll say something like when the amylase fits into the when the starch fits into the amylase, the active site of the amylase, an enzyme substrate complex is formed. Okay, that's the ESC. Then I end it off with something called SPU. SPU, it stands for substrate, products, and unchanged. So I will start by saying the substrate is broken down into its products, which are maltose. Okay, in this case it will be maltose because it's from starch. Then I'll end it off by saying the enzyme remains chemically unchanged and it can catalyze another reaction. Okay, so that's how I'll write my lock and key. So remember the formula. The formula is L and K first. Tell me the lock and the key. Then SAC. S stands for the specific shape, specific 3-dimensional shape, and the A stands for active site, and the C stands for complementary. Then the ESC stands for enzyme substrate complex, and the SPU, which is substrate, products, and unchanged. Okay, so if you can kind of like keep this formula in your brain, as you write it out, you'll be able to create that checklist at the side, and you'll be able to test and be very certain that you got all the points for this lock and key kind of hypothesis. Okay, so I want to quickly talk about the characteristics of enzymes. Okay, we've already covered a little bit of it, and so we just zoom through this. Okay, enzymes speed up chemical reactions. Enzymes alter the rate of chemical reactions that are in the cell. Enzymes speed up the chemical reactions by lowering the activation energy needed to start a reaction. Enzymes are specific in action. Enzymes have active sites. Only substrates with a shape complementary to the active site can fit into the enzyme. When an enzyme binds to its substrate, it forms an enzyme substrate complex. However, high temperature, acid, and alkalis can affect the shape of an enzyme and consequently its function. Enzymes are required in minute amounts and remain unchanged at the end of reaction. The word minute means very little, not minute. Okay, not minute. Enzymes are very efficient molecules. So, since they remain unchanged in the reactions they catalyze, the same enzyme molecules can be used over and over again. Thus, a small amount of enzyme can catalyze the reaction for a large amount of substrate. For example, just a spoonful of washing powder contains enzymes is enough to wash a load of clothing. Okay. Enzymes are affected by temperature. Okay, so this is something that we will start to focus on. Okay, the above one we already talked already. So remember the lesson objective, right? We're supposed to learn that the effect of temperature on enzymes. Okay, so these two things are temperature and pH, very important. Okay, here we have a graph of the rate of enzyme activity or rate of reaction over a certain temperature. On the left hand side, it'll be 0 degrees, and on the right hand side, it'll be, let's say, 100 degrees. And some point will be the optimum temperature. Okay, so let's go starting from the first step from the left. Okay, so for how enzymes are affected by temperature, usually I will go from the left all the way to the right. Okay, so part 1. An enzyme is less active at low temperature. Okay, or you can say the enzyme is inactive at low temperature. So at low temperature, the kinetic energy of molecules is low. Enzyme and substrates molecules move slowly. The rate of substrate molecules colliding with the enzyme is low. Now you don't really have to write all this. Or rather, it's not the most important right now, but just think about it in terms of your chemistry, which you probably have learned already, right? So this is your enzyme, these are your substrates. Okay, if there's very little kinetic energy, which is basically heat energy also, right? You know, when there's heat, there's kinetic energy. So they're moving very slowly, and there's very little chance of them, you know, touching each other and bumping each other, because there's too little kinetic energy. They can't move. But as the temperature increases, the rate of enzyme reaction increases, because increasing the temperature increases the kinetic energy of molecules. The rate of effective collision, which is the rate of substrate molecules colliding and fitting into the active site of the enzyme, is increased. This increases the rate of enzyme substrate complex formation. Okay, so what happens is that as it gets more, the heat increases, right? The temperature increases. Now the enzyme starts to move more quickly. The substrate also moves more quickly, and they can collide into each other. When they collide and they angle, you know, into that perfect fit into the active site, that is where that's why I say as an effective collision. The substrate fits into the active site, and so there can be a enzyme substrate complex being formed. So that's at number 2. That's why you see it's going up. Do be aware, the shape of this graph is important. Don't just anyhow draw one mountain. It is like a, how to say, like a pushed forward mountain, or like a roller coaster, right? You slowly go up the roller coaster, then you zoom down the coaster. So if you to ever draw this graph, make sure it looks exactly like this shape. Okay. Alright. Now at part 3, this is what I call as the optimum temperature, where the enzyme is most active. So why is it the most active? It's because each enzyme has a specific optimum temperature that they like. You know, they are not alive, okay? The enzymes have no mind of its own, but it's just that that's the temperature that they work the best at. Okay. Now for humans have a body temperature, right? It's actually, our body temperature is about 37 or so. Okay, so our enzymes actually work best near to our body temperature, which is actually about 40. Okay, just slightly above our body temperature. I would think 45 is just a little bit high. Okay, I don't think that, maybe I'm wrong, maybe that this person knows it better, but I would say 37 to 40 is the optimum temperature for humans. Okay. Alright, so that's the point at number 3. And please be aware that there's other animals out there, right? Not only humans. So for example, there are some bacteria that could be living in volcanoes. They might have a very high optimum temperature. There's some other things that could be living in cold areas, so their optimum temperature might be lower. So this is just a gauge. Every enzyme in every creature will have their own optimum temperature. Okay, so you need to be aware of how to read the graph also, and see the point, which is the highest point, and then read it on the graph. Okay, now at point number 4. Okay, now you see that the rate of reaction starts to decrease. It's going down. Why is it going down? Because when you increase the temperature above the optimum, it causes a rapid decrease in the rate of enzyme reaction. This is because the active site of the enzyme molecule begins to lose its original shape and is no longer complementary to the shape of substrate molecules. Now this is what is termed as denaturation over here. Denaturation. Okay. So point number 5, at this temperature, all the enzyme molecules have been denatured, or you can say most of the enzymes are denatured. So that's what's happening here. Almost all the enzymes have been denatured, and so there can be no enzyme activity. So what is denaturation? Denaturation is the change in the 3-dimensional structure of an enzyme or any other soluble protein caused by heat or chemicals such as acid and alkali. Okay, so there are 3 things that cause denaturation: high heat, extreme acidity, and extreme alkalinity. Okay, so let's look at the picture of denaturation. Denaturation is the loss or alteration of the enzyme's active site. The substrate can no longer fit into the enzyme's active site, and no reaction will occur. Hence, when an enzyme is denatured, it can no longer act as a catalyst. So this is a good, proper enzyme, right, with a specific 3-dimensional active site. And so our substrate molecule can fit in very nicely. Okay, but if you start to heat it up, protein would react to the heat, Alright, and as it's being heated up, the shape of the protein will start to change, and it changes into this funny shape. Now, the substrate can no longer bind to it because there's no more active site. Active site is lost. Okay, and this happens the moment it crosses the optimum temperature. So even at this point, 0.4, some of the enzymes is already starting to get denatured already. And eventually, all the enzymes will be denatured at 0.5 or most. Okay, so just a few things. You cannot say that the enzyme die. Enzyme got no life. Enzyme are not living things. Cannot say enzyme die. Okay, try to avoid saying enzyme are destroyed also. That's not the very scientific word. Okay, so if you can, you must, oh wait, not you can, you must say enzymes are denatured. Okay, you cannot say that for cold temperatures though. Okay, so remember at cold temperature, like maybe 4 degrees, 1 degrees, 0 degrees, the enzyme is inactive. It's not denatured. So this point, don't say it's denatured, is inactive. You can actually thaw it out. Thaw means that you melt it out, and then slowly from the frozen enzyme, right, it can slowly not revive, I saying the wrong thing. It can slowly become more active, and then it can be used again. Okay, in fact, most of the time when you purchase the enzymes from the supplier, they come frozen, and they come to you, then you have to slowly, not too fast, but slowly warm it up until it's usable again. Okay, so I want you to try this. Well, you can't try this, it's a lab experiment. Okay, we are going to pause here, and then we'll continue to talk about the effects of pH on enzymes in the next lesson. Okay, see ya. Peace out.